Thank you, Maeve. Thank you, Hannah. Thank you, Notan, for giving me this opportunity to interact with colleagues all around the globe. I'm just going to talk a bit more about a recent publication from this June. It was online from March, but it will be published in June in official format, which states Middle East Respiratory Syndrome, Coronavirus, MERS-CoV, a 10-year, because it's almost 10 years, 2012 since the first outbreak, a global analysis of human and camel infections, and to look a bit deep more into genomic sequences, lineages, and the geographical origins, how well this has been disseminated and distributed in terms of the transmission dynamics of this particular virus, which is equally a coronavirus family, such as the one which we have faced, like the 2019 novel coronavirus, which is later translated into the SARS-CoV-2, which causes the disease, the COVID-19. To begin with, well, I will start with the overall introduction, a bit more about coronaviruses, a bit about MERS-CoV in terms of diagnosis, symptoms, but also in terms of treatment, vaccines, and other, like other characteristics. Then dive a bit deep into the paper so that you have an overview of the MERS-CoV in as a whole. So coronaviruses belongs to the family Coronaviridae, which is a positive single-stranded large RNA viruses. It is known to infect humans, but also it is known to infect a wide range of vertebrates, which includes amphibians, birds, and other mammals, which includes bats. The morphology is based on spherical virions, which has a core shell and has surface projections. This is how the name was coined, like it looks like determined like a solar corona. In Latin, corona means like a crown. That's how the name corona came into existence in terms of coronaviruses. The genome size varies between anything between 28,000 base pairs to 30 to 32,000 base pairs, kilobases. As known, any given virus has to be coded by different proteins. So obviously these proteins are coded by genes. You have 4 structural genes like in SARS-CoV-2, which is the nucleocapsid protein, the spike protein, a small membrane protein, and a membrane glycoprotein. And in addition, there is some coronaviruses which also has membrane glycoprotein. Just to move forward, the very first discovery of the coronavirus happened in 1966. What happened in 1966 is there are 4 colleagues, Tyrrell and Binu, and Hamry and Prochnow. These are the colleagues who have discovered the coronaviruses. What they did is, as you see, these are the 2 different articles which first described the coronaviruses in 1966. What they did is they started to collect, especially the Tyrrell and Binu colleagues started to collect it between 1961 to 64. They collected the nasal washings of the adult and children individuals who have flu-like symptoms. And what they did is they tried to collect these nasal washings and try to culture them in different medium, including embryonic cell lines and tracheal organ cultures, and so on and so forth. What happened is Tyrrell and Binu found that they could passage a virus, which was first termed as B814. And this is the very first terminology of the virus of coronavirus, B814, which was found in the human embryonic tracheal organ cultures, which are obtained from the respiratory tract of an adult infected with common cold. As you may see, between 1961 to 64, they were using the staff members and their families to collect the nasal washings. And what they did was, once they can able to culture these B814 viruses in organ cultures, they try to demonstrate that this is an infectious agent by inoculating the medium from the cultures intranasally to the human volunteers. It's like a human challenge infection model, that the colds that were produced after the intranasal inoculation is due to the virus, which is from the organ cultures, which is being cultured. However, there was a problem that these colleagues could not manage to grow this virus because of this unusual property of this tissue culture from samples obtained from the medical students. At the same time, there were colleagues from Hamry and Prochnow. They both were able to grow this virus in a tissue culture from the medical students who had cold infections. What happened is they could clearly state that this virus was ether sensitive. Therefore, they call this virus as 229E, which is the ether sensitive version. Equally, at the same time, Tirir and Almeida, they performed electron microscopy on the fluids from the organ cultures infected with B814, which is the predominant or the focal virus, where they could show that this viral particles resembled infectious, similar to the bronchitis virus, similar to that of the chickens. This is the very first discovery of the coronaviruses that happened in 1966. And coronaviruses are belonging to the Riboviria from the Orthonoviriae family, from Nidovirales order, from the family of Coronaviridae. So far, what we know is there are 4 subfamilies from coronaviruses, namely the alpha coronaviruses, beta coronaviruses, gamma coronaviruses, and the delta coronaviruses. This is not related to the SARS-CoV-2 alpha, beta, gamma, or the delta variant. These are the different types of coronaviruses, which means the coronaviruses alpha and beta originate from mammals, especially from bats, whereas gamma and delta originate from pork, from pigs, as well as from apes, from the birds. So far, we know that there are 7 different subtypes of coronaviruses that are known to infect humans. Among these alpha, beta, gamma, and delta, beta coronaviruses are the most lethal, which causes the severe disease, which leads to the fatalities. As I said, there are 7 different coronaviruses. 4 of these coronaviruses that are known to infect humans are generally mild. As you see from the slides, there are 2 of the beta coronaviruses, there are 2 of the alpha coronaviruses. And these OC43, HKU1, 229E, and LL63, these continuously circulate in human population, both in adults and in children population worldwide, with their seasonal incidents occurring explicitly during the winter months, especially in temperate climate regions. These 4 causes mild disease episodes, whereas the other 3 human coronaviruses that are known to be more severe and fatal are caused by 3 different human coronaviruses, and all these 3 belong to the beta coronaviruses family. The first one being the severe acute respiratory syndrome coronaviruses, the very first ancestral SARS-CoV-1 or SARS-CoV, which is a beta coronaviruses, identified first in 2003 from a patient from Hong Kong, identified mostly in Southeast Asian region, bearing the large morbidity and mortality rates, followed by the Middle East respiratory syndrome related coronaviruses, the MERS-CoV, which is also a beta coronavirus, which is identified in 2012, followed by the latest one, which we have experienced in the last 3.5 years, which is the severe acute respiratory syndrome coronaviruses, the 2019 novel coronaviruses, which is renamed as SARS-CoV-2, which is a beta coronavirus, which causes the COVID-19, the disease, which is identified in 2019. If you look at the timeline of the coronaviruses, the OC43 and the 229E was first discovered in 1966, as I have explained regarding the discovery. The OC number comes from, the OC acronym comes from organ culture, and 229E, it's not 299E, it was a typo in the paper. I found it out. So it's 229E, which is the ether sensitive, was the one which was discovered in 1963. And then 2003, we have the very first SARS-CoV-1 outbreak from the Southeast Asian region, which has led to 774 deaths with 8,098 infected patients, followed by a strain in 2004, which is an alpha, HCOV-NL63 in 2004. NL is the one which stands for Netherlands, which was first identified in the Netherlands strain, followed by 2005, HCOV, which is a strain from the Hong Kong, HKU1. And then came the 2012, which is the Mediterranean Respiratory Syndrome coronaviruses 2, which had the highest fatality rate among any given coronaviruses, which had 35% case fatality rate. Followed by the 2019-2020 NCOV, or the SARS-CoV-2, which has been the large pandemic in the last 2.5 to 3 years time. But then now, since you may know, like in the last 2 days, WHO has reduced the involvement of SARS-CoV-2. It's no more a public health of emergency concern. What is to be noted is here is most of these coronaviruses, especially the 229E or the OC43 or the NL63, are often we have been exposed to these viruses, especially the children population. And these are often co-detected with other respiratory viruses, particularly with the respiratory syncytial virus RSV, which we call it as. Coming back to one specific form of the beta coronaviruses among the three, which is known to be fatal, the SARS-CoV, the MERS, and the COVID-19, which is caused by the SARS-CoV-2. We are now going to look a bit more deep into the Middle East Respiratory Syndrome coronaviruses, MERS-CoV, which is basically a viral respiratory disease caused by the virus of coronaviruses, which is a beta coronaviruses. This virus was very first identified in Jeddah in Saudi Arabia in June 2012. The very first case was in June 2012, and there was a physician who was working at the UAE hospital in Jeddah. He was an Egyptian physician, Ali Muhammad Zaki. He was the first one who discovered, or who first discovered this very first case, which is similar to flu-like symptoms, similar to SARS-CoV-like 2003-like symptoms. And the incubation period, which means the onset of infection, starts between 2 to 14 days with a mean infection time of 5 to 6 days. Starting with, obviously, as you may know, as you might have experienced, few of you guys, like fever, starting with a fever, followed by a cough, dyspnea, shortness of breath, and possibly there are also cases with diarrhea as well. This was the very first case which was identified in Saudi Arabia in June 2012, but the very second case was proven case, or a laboratory proven case, was identified in London in September 2012, and they called this as London Novel Coronavirus 2012. As we have said, like 2019 novel coronaviruses, they have said this as London 1 Novel Coronaviruses 2012. Obviously, pneumonia, which is a predominant feature of a coronaviruses, which is very common, but not that all MERS persons will develop this condition. MERS-CoV is known or observed to be a zoonotic viruses, which is very linked to human infection, explicitly coming out from the dromedary camels. If you look at the geographical distribution of these viruses in terms of epidemiology, they are mostly predominant in Middle East, in Africa, and equally from 2015 from South Asia. If you look into the transmission patterns of MERS-CoV, so MERS-CoV is potentially transmitted by the bats, The very first cases were linked to the Egyptian tomb bat from the family of Abelonoreidae, which is the sheet-tailed bats. But equally, most of the families of simple-nosed bats, such as the Vespertilionidae, is equally known to cause infection in humans, but also in dromedary camels. What is known is the source or the origin is being the bats, the intermediate hosts to be the dromedary camels. There are two types of camel population. One is the dromedary camels, which you see just like a one hump in the back. There is also the other camels called the bacterian camels, which has two humps, 1 and 2, which means it looks like a B in the alphabet B, so it has two humps. So the intermediate host here is very specifically dromaeosaurian camels, and the zoonotic transmission happens when there is an interaction between the infected person as well as not the infected person, the person being exposed to the infected camel. And most of these cases comes from camels, as you see. Therefore, you may ask what might be the reason. In Saudi Arabia, people do eat camel meat, milk, camel milk, and equally, there is also people who drink camel urine. There are recommendations that people should eat fully cooked camel meat, pasteurized camel milk, and avoid drinking camel urine. What is equally known in terms of transmission is a study between 2010 and 2013 during the outbreak and after the outbreak can clearly show that many of these many of these dromedary camels have revealed high neutralizing titers for the antibodies to the MERS-CoV-2 in the serum. There was a study which has looked into 310 or 320 of these camels from dromedary camels, which looked into different populations where they could clearly show that they have enough neutralizing antibodies which are being produced in the body against the MERS-CoV. It's not about, we do have a kind of a feeling that we always check how much antibodies we do have in terms of, an effective response against an infection. It's not about the amount of total antibodies that we might have after a vaccination. It's about how much of the neutralizing antibodies that we have, that we might have to protect against an infection. That's how it works. And the WHO, even for the COVID-19, has re-strategized the efficacy of the COVID-19 vaccine is not based on infection after the vaccination. It depends now based on the neutralizing antibodies that an individual produces. Yeah, so to keep it short, most of the transmission has occurred in close contact with severely ill patients in healthcare or in household settings. As you may see, many of these respiratory infections have happened mostly in the healthcare associated transmission or within the household. But if you look into the community transmission, there is far little known in terms of community transmission. If you look a bit more deep into the cluster size in terms of infections, the cluster size ranged from, like between 1 to 26 people, that one person can infect anywhere between 1 to 26 persons with an average of 3 persons that he could transmit the viruses. So that's what we know in terms of the transmission dynamics. Most of these viruses are healthcare or household transmission, not much in terms of community. How do we define a probable case? A probable case is defined by a fever, followed by a pneumonia, or a history of travel from other countries from Arabian Peninsula within 14 days before symptom onset, or a close contact with a symptomatic traveler, or a close contact with a confirmed MERS case while the case was ill. This is how a case definition is. And if you look at the MERS situation update in 2023, which is the recent report from last week, we have 2,604 laboratory confirmed cases with 36% of case fatality rate, which means 936 deaths. And the last death due to MERS-CoV-2 occurred in 2023 in Saudi Arabia. If you look at the fatality distribution of the cases, you have primary cases and you have secondary cases. Primary cases is with direct contact with the source of origin, and secondary cases is the contact with the primary cases. If you look at the primary cases, the age group of 50 to 59 has the highest risk, whereas the age group of 30 to 39 years old had the highest risk in terms of the secondary cases. When we look into MERS cases in Germany, Germany has reported 3 imported cases, one in 2012 from a returning traveler from Qatar who has recovered, who is 45 years old. The second case in March 2013, who was also a returning traveler from Abu Dhabi who died, who was 73 years because of comorbidities. And the third person is also in February 2015, who was also recovered, who was 65 years old. But there are always sporadic cases of the MERS-CoV, which is happening all around in especially in the Middle East. The last cases which is being reported is in Oman and Saudi Arabia in the last years. The MERS outbreak is not only restricted to the Middle East. There was the very first importation of the Middle East case outside the Middle East Asia, which has led to the cases in South Korea, which is a 68-year-old man who flew in from Kuwait to who flew in from Saudi Arabia to Seoul, who was the index case for the South Korean outbreak, which has led to 186 confirmed cases in Korea, 185 in Korea and 1 in China, which has led to 38 different deaths. But even though there are in later stages, there were in 2018, there were sporadic cases, but then these are well quarantined so that the spread of the virus has been well contained. If you look into the MERS-CoV-2 genome, obviously you may know, we might have heard about all these different types of SARS-CoV-2 genome. It's similar to that. You have the open reading frame, you have the spike protein, you have the envelope protein, you have the membrane protein, you have the nucleocapsid protein. As you know, for the SARS-CoV-2, we have the ACE2, which is the angiotensin converting enzyme 2, which is the receptor in the host cell from the lung alveolar epithelial cells. Therefore, the binding of the spike protein of the receptor binding domain to the ACE2 establishes the infection in and lung alveolar epithelial cells, which is in case of SARS-CoV-2. Whereas in the MERS-CoV, we have the receptor binding domain is the dipeptidyl peptidase 4, which is called DPP4, which is predominantly conserved in humans, but very variable within camels, which means camels are able to recombine these virus particles over a period of time, whereas there is little evidence in terms of recombination events in terms of the humans. If you look into the diagnostics, you have a real-time PCR-based diagnostics. WHO recommends you have to do for 2 specific genomic targets, even a positive result for 2 specific genomic targets being positive, or if you can look for a single positive target, but subsequently it has to be confirmed by a sequencing by a second PCR, by a second sequencing of a second target. Serological tests are used, but these are only recommended for surveillance and not used for any given diagnostic purposes. In terms of vaccines, what is known in terms of MERS-CoV? We have plentiful of vaccines which are in pipeline at this moment. The first one is the Gamaleya, which is similar to the Sputnik, what you might have heard about the SARS-CoV. It is a similar one, which is a phase 1, phase 2 studies from the Gamaleya Institute, which also produced the Sputnik vaccines. The other one is the Chadux, which is the Oxford based on the adenovirus vector-based platform. The third one is the modified Vaccinia Ankara vector-based platform, which is produced by a company called IDT Biologica from Dessau in Germany. And they are doing the clinical trials with CEPI, Coalition for Epidemic Preparedness Initiative, across Saudi Arabia. There are other vaccines such as the Intravac from NL, which is also running the CEPI trial, which is looking for a broader protection for SARS-CoV-1, CoV-2, and MERS-CoV. And the other ones from Novavax are currently being paused at the preclinical processes. just jump into the study. What we wish to do in this study is, of course, in last 10 years, we would like to know what has happened in terms of shift of these genomic sequences, if there is a trend that we would like to see in terms of the geographical origins between 2012 and 2022. Is there a recombination happening between the viral lineages? Is there a different clades? Is there a specific geographical domain where specific lineages are represented? Is that so that we would like to know if the animals being there, obviously the reservoir for the MERS-CoV-2 infection, but obviously we would like to know if there is also recombination happening in terms of the humans. Why the infection is very well contained so far, but there is some sporadic outbreaks in the last 10 years time frame. So what we did is, so we looked at all the different search terms, used the different databases, look for the different search terms, look for all the different sequences. And what we did is, so far, there were 728 different genomic sequences so far deposited for the MERS-CoV-2 genomes. What we did was we extracted 4,061 bases of the S gene, which is predominantly having the receptor binding domain, which is known for different variations. And we looked for the viral dynamics and the transmission patterns. So I would like to give you a small, brief summary before we dive deep into the results. There are two terminologies which you will be exposed. One is called lineages, the other is called clade. The lineages are basically the relationships through time between the ancestors, whereas the clade is basically the branched monophyletic group containing a single ancestor. I hope you understand. In a worst case scenario, I could explain you in a bit more in detail that we know that we all come from apes. So the apes becomes the lineage, whereas the clades, or we are the descendants of the different types of apes. Yeah. So this is how it is called as. To keep it more simple, I can give you another analogy. Let's say Mozart. Mozart had 6 children. None of them had children on their own, which means the composer's lineage ended after one generation. Yeah, which means there is only one clade and one lineage with the children, and it's ended. Clade is a branch which includes a single common ancestor and all the descendants from the clade. Let's move down to the results. What we did is we looked at 501 of the 728 sequences, which are the full-length genomes of the MERS-CoV. As you may see, we have most of the sequences come from the Saudi Arabian Peninsula, but also from the United Arab Emirates, partially from Jordan and the South Korean clusters, but equally other samples are also coming from other zoonotic origins, such as the Camellias, dromedarius species. So these 501 sequences were subjected to phylogenetic sequencing. But if you look at the different years distribution, the most of the sequences which were deposited from the global databases come from 2015, 2016, and 2014. Among these sequences, 226 of them come from infections in humans, whereas equally 265 of them come from the camels as well as from the llama population. What we observed is we had 3 different clades: clade A, clade B, and clade C, as you may see, whereas clade A and clade B are early cases of the MERS-CoV, which is found in 2012, whereas the clade B is the one which are genetically different from clade A and clade B, which means these have the whole clade B has evolved over the last 10 years time frame. Okay, this is clade B, which means the geographical origin or the ancestral origin of clade B has evolved from clade A and clade C, which is the ancestral ones. Among this clade A and clade B, we have 6 different lineages, 1 to 6, from 1 to 7 from clade B, which all belong to the clade B. So the clade B is the most predominant clade which is currently in circulation with all these 7 different lineages. What is interesting here to see is most of the clade A and clade B, clade A and clade C belongs to the earlier cases in 2012, whereas the clade C belongs to the zoonotic camels transmission only from Africa, which means any African dromedary camels has clade C, whereas the clade A belongs to the camels, but also equally the virus isolated from the humans has the clade A during the 2012. Whereas the clade B is the one which is predominantly right now being circulating in the human population, but also in the camel populations per se. But equally also, if you look into the spike protein per se, if you look at the spike protein, there are different domains: N-terminal domain, C-terminal domain, receptor binding domain, loop domain, histone-rich domain, CP, and so on and so forth. The clade B was the predominant one, which was responsible for the South Korean outbreak in 2015. But equally, if you look at different clades, there are only 3 clades: clade A, clade B, and clade C. Clade A and C are extinct, which means they are now considered as non-circulating strains, whereas clade B has evolved over time, over space, both in humans as well as in dromedary camels, which has led to even 5 lineages to 7 different lineages, which means the recombination rates within the clade B has occurred more often. It appears that despite a steady import of camels, even from Africa to Southeast Asia, there is no any recombination or no real infection happening from the African camels in Arabian camels. So what does this mean? Clade A is found in 2012, both in humans as well as in camel population in Saudi Arabia, whereas clade C is very defined to the African camels, whereas the clade B is the one which is circulating with many different lineages between camels and among the humans, which clearly shows and explains as the Saudi Arabian MERS-CoV strains are completely different from the African MERS-CoV-2 clades. The next point is in terms of lineages. As I said, there are 7 lineages, 5 lineages in 2016 to 7 lineages in 2017. And these lineages all belong to only clade B, which means the clade B is the most predominant circulating strain, which is evolving over time, explicitly only in camel populations, because the lineage 1, lineage 2, and lineage 3 are of no interest. Lineage 4 is very critically important in terms of the outbreak during the Hajj pilgrimage in 2014, whereas the lineage 5 is a recombination, which means 2 viruses infecting one single host cell, leads to the recombination of lineages 3 and 4, which leads to a new lineage of 5, which is equally being important and being responsible for the outbreak, what has happened in Southeast Asia or in South Asia, especially in South Korea. Whereas the other 2 lineages, lineage 6 and 7, are very specific only to the camel populations. Yeah. Taken together, what we could say is the number of human MERS-CoV cases have declined in the past 5 years. And there is not much variants continue to circulate in the humans, but the MERS-CoV variants in terms of different lineages are continuing to circulate in the camel population. not in humans. Several of these variants have appeared to occur in the camel population, but very less frequent in humans over the time. The recombination rates of these viruses, especially the MERS-CoV, clearly indicate that multiple lineages are occurring most predominantly in the zoonotic population, which means camels have an adaptive potential to have the recombination rates, which means they are the capable enough of spreading the new variants or the new lineages. Therefore, a continuous surveillance and equally genomic analysis of MERS-CoV-2 infections, especially not only in humans, but most importantly in camel population, is very important to drive this in terms of understanding the endemic and epidemic transmission potential of the MERS-CoV-2 viruses. And equally, the WHO blueprint list of pathogens shows that the MERS-CoV is a global public health security, which requires more in terms of research, in terms of surveillance, in terms of new diagnostics, in terms of new specific treatments, equally in terms of vaccines that could provide a basis to infection control. Thus, the spread of the virus is contained, thus the elimination and eradication of this virus. With this, I would like to thank you for your attention, and I'm very happy to take any given questions and open for the discussions. Thank you very much.